R&D department. Sir, can we launch souvenir?
Good afternoon, everyone. My name is Ahmed Irshad, and I am from Wales Institute of Science, Technology, and Advanced Studies. And
Wait a
this,
second. Wait a minute. Wait a minute, Amma.
Wait a minute.
tell me.
Hello.
Yeah, ma'am.
Ashwad, wait a minute.
Tell me.
Wait a minute. We will announce you, then you start the session. Okay? Just wait. Shyam, follow.
So, participants, all of you are seeing that we have released our souvenir. So this is the first page, okay, front page of our souvenir, and this is a message from the chief editor, Mr. R. John Sunil. This is the message received for our PHARMA Analytica 2020 from the Vice Chancellor, Sri Jayesh Ranjan, is IAS, Jawaharlal Nehru Technological University. Next, we have also received the message registered Dr. A. M. Mansoor Hussain from Jawaharlal Nehru Technological University. We have also received the message from Dr. G. Krishna Mohan, Professor Pharmacy and Director of Institute of Science and Technology, JNU, Hyderabad. Next message we received from the Dr. T. Narayana, President IPA, the Indian Pharmaceutical Association. So this is the message from our Chief Patron, Dr. G. Srinivas Reddy Garu, General Secretary, Sri Vageshwari Educational Society. This is the message we received from our Dr. R. Ramakrishna, Principal and convener of this e-conference. So now, so this is the organizing committee. This is a scientific committee. This is the steering committee. This is the executive committee. This is the press and media committee. And this is about the details about the e-conference. And this is about our college. And these are the acknowledgements.
Thank you, sir. Dr. T.G.K. Murthy, Principal, College of Pharmacy. And please
Good afternoon, one and all. Myself, Dr. D. Shravan Kumar Doli, Associate Professor, Department of Pharmacology. Let me congratulate all the delegates who have submitted their abstracts of their research and review from the different departments of the pharmacy. Scientific committee headed by me and other committee members have gone through the abstracts, and we selected the few number of the abstracts for the presentation, and the participants are requested to present their presentation as per their allotted slot. The slot already we sent by mail and message. The duration for the presentation is about only 10 minutes and followed by the evaluation of the presentation by subject expert, Dr. Rajendra Prasad sir, Director R&D Department, Viper, and Dr. T.G.K. Murthy sir, Principal, Babarla College of Pharmacy. And the names of 5 best participants will be announced during the voluntary session on the second day of this conference. And kindly mute your audio while the session is going on, except the presenter to avoid this technical errors. May I now take my privilege to invite first participant, Mr. Ishrant, to present their presentation.
Thank you, sir. That will be me. So, good afternoon, everyone. I hope everyone is safe and sound. So, I'll share my screen. So, my name is Hamad Irshad, and I'm from Wales University. So, can everyone hear me properly?
Yes, you can go.
Okay, thank you. Okay, so this is my topic. My topic is efficacy of aspirin in cardiovascular system. So, and I'm presenting for PHARMA ANALYTICA 2020. And about aspirin, we know that aspirin has been used for the treatment and prophylaxis for cardiovascular disease in its normal dose and in its low dose. So the clinical benefits of the drug in the primary prevention of cardiovascular diseases is proven from current evidences, its benefits and
Please,
effects on the secondary prevention of the disease is not well established and
remaining
unclear.
participants, please keep
So
mute.
current recommendation and guidelines on the use of aspirin for secondary
Please
prevention is
mute remaining participants.
widely varied and have conflicting statements at times. So this is believed to be due to the interpretation of results from various clinical trials. So the main theory that goes around is that aspirin, due to inadequate suppression of COX-1, unlike the traditional NSAIDs and COX-2 inhibitors, it just inhibits COX-1 in conjunction with COX-2, so which leads to aspirin not necessarily reducing the risk of atherothrombotic complications and might actually lead to increasing it. So the concern regarding using aspirin in the treatment in the prevention of cardiovascular diseases is that the main side effect is bleeding, and predominantly in the upper gastrointestinal tract, and rarely involves intracranial hemorrhage. So doses above 30-day increase the risk by inhibiting COX-1 activity and thromboxane A2 production in platelets, thereby reducing platelet aggregation and interfering with primary hemostasis. So dosage levels of 75 to 325 mg per day approximately doubles the risk of major intestinal bleeding from aspirin treatment, and risk of bleeding increases with age. Also, the use of co-medications such as NSAIDs appear to modify bleeding risk with low-dose aspirin. Bleeding risk factors could potentially have a large impact on the absolute number of excess cases of bleeding and therefore net benefit consideration. So, low dose and normal dose aspirin are recommended for the prevention of cardiovascular diseases, and the drug has been also reported to be directly or indirectly involved in precipitating CVDs like intracranial bleeding and gastrointestinal hemorrhage, and also myocardial infarction, on top of the side effects that I've already explained. So the problem is that long-term effect of the drug being that it is causing the disease that it is bound to prevent. So that is the main problem with using aspirin as a preventive drug for cardiovascular complications. So right now, these are the guidelines for low-dose aspirin in use for cardiovascular prevention. As we can see, it has been updated till to 2019 by ADA and ACC. So these are heart associations and diabetic association. These have updated their guidelines, and till today, this has been followed for prevention of cardiovascular disease if they are found to be having risk. So the problem we are facing right now is that the drug causes the disease that it is bound to prevent. So apart from all this, it has side effects, and beyond that, there is this new term called aspirin resistance. It is not like antibiotic resistance where a single strain is unable to react or something. It's just used when drug aspirin is unable to produce a response to inhibit thromboxane A2 in vivo or to protect the patient from the complications. So what happens is the drug does not cause any response in the patient anymore because of taking the drug frequently in low doses for a long time, which is what the guideline says. So the term does resistance does not mean inter-individual response to a treatment or the drug, and is quite often deceiving and is actually a phenomenon that can be quantified, which has a direct effect on clinical efficacy, leading to the changes in the antiplatelet therapy. So it is basically how the drug reacts, how the body reacts to the drug. So, however, the relation between platelet activation in vivo of the ex vivo functional indices of platelet capacities is highly unknown, and different tests of aspirin responsiveness do not correlate and correlate poorly when they actually do so. So, though aspirin is believed to be the conclusion, I would like to conclude this paper by saying that it is believed to be cardioprotective and is used for cardiovascular protection and as prophylaxis, but its potential benefits don't outweigh its harmful effects. So it is better to consider the use of the drug for acute conditions rather than its prescription for a long term, as the effects of a drug being taken long term might have additional conditions added to prevailing conditions due to the use of the drug. So what happens is we are giving the drug to a patient who is having a risk for cardiovascular complications. So if we give a drug that might also cause the same thing, its potential benefit is not outweighing its effects. So it's better to avoid the drug, and which might lead me to say that there must be more studies conducted on the drug to prove its effectiveness as a preventive measure for cardiovascular diseases. So this necessitates the implication of monitoring systems and other clinical regulations that might help detect a CVD and events that might unfold in the future. This can be avoided by better adherence and compliance by the patient and involvement by the patient. So that's basically it, and these are the references that I had to go through for this course. Thank you. Doubts and questions are welcome.
Thank you, Mr. Ishant, for your very good presentation.
Thank you, sir.
Now I humbly request subject expert, Dr. Rajendra Prasad, sir.
Ishant, are you here?
Yeah, I am here.
Yeah, good presentation. And as we know that the aspirin is a very well-known drug for the cardiovascular, and it's also available at the cheaper cost when compared to others. So, any other alternative drug in terms of activity and in terms of the availability of the for this aspirin?
As of now, there is no any proved drug that could be used as a secondary prevention. That's the problem we have. This is the most known drug to humankind that we've been using for a very long time. So the problem is we don't have any drugs that can be used for a long term. That is why I was insisting on doing studies, long-term studies for the prevention of cardiovascular complications in a long term.
Okay. And you are talking about resistance of the drug aspirin. So there is any relationship between the resistance and the drug dosage?
No, the actually it's not the dosage, but the frequency of the drug and the usage of the drug in a long term. So what happens is when we use it for a long time, the drug does not elicit any response after a long time, because your body and cells are used to the drugs normal dose. So we have to up the dose so that we could use it. If we up the dose, it would change the effect of the drug, which is NSAID and not an antiplatelet drug. So that is the problem it's posing. So which makes us be redundant about the use of the drug to to have a wide response in the usage of the drug, and studying the drug for further complications it might precipitate.
Okay, good luck. Thank you.
Okay.
Thank you, sir. Next, now I request evaluate Dr. M. Srinivas Reddy garu to evaluate this presentation.
Hello.
Yes, sir.
Hello.
Yes, sir.
Yeah.
I'm here.
As per Ranjit Professor are asking questions. Okay, your presentation is very nice presentation.
Thank you, sir.
But why you are choosing the aspirin resistance mode?
Sir?
Only your I coming on last slide only. Please open your first two slides.
Okay, sir.
Hello.
I'm here, sir.
Hello.
Yeah, I'm here. Hello?
Hello.
Can you hear me?
Yes, sir. He's in
Hello.
Mumbai.
I'm here.
Yes, sir. Okay. Yes, sir. He is online.
Srinu, sir. Unmute yourself, sir.
Hello.
I'm here.
Yeah, it is a COX-2 inhibitor.
Yes, sir.
What is the mechanism action of NS side? Other than aspirin, any drug can be used for this?
Yeah, there are many NSAIDs known, sir, like ibuprofen.
Oh, okay.
Okay, sir. So it's not NSAID that we are that aspect of the drug is not focused. So it's the secondary prevention that has been used in lower doses. When used in lower doses, it's been used for
Lower dose means, for example, in infants.
no, not like that. When in older patients, sir, in patients
Only
who are
older patients.
posing
Okay.
who are posing risk for cardiovascular complications, there are guidelines actually. There are which I've should I show that too? So these are the guidelines that actually encourage to use the drug aspirin for patients who are posing the risk for bleeding and other cardiovascular complications. So the problem is that the drug might actually cause the actual disease we are trying to stop. The
Okay.
NSAID aspect of the drug is not focused. It's that's another thing. We have a lot of new NSAIDs in the market, and we are using it right now. Aspirin is not used as NSAID anymore. I think so. In my
Exactly. Thank you.
oh, thank you, sir. It's my pleasure.
Thank you, sir. Thank you, sir. Thank you, Mr. Ishant.
Thank you, sir.
Next presentation. Next one, Dr. Deepika Raina, Assistant Professor, Chiktara University, Punjab.
Chithara.
Dr. Deepika. She was absent. Okay. Good. Next, Sony, PG student, Sri Krupa Institute of Pharmacosciences, Siddipet. Sony, did you hear me?
Yes,
Sony.
sir, yes, sir. Yes, sir.
Okay, go for your presentation.
Okay, sir.
Sony, are you unable to share the PDF? We are ready to share the PDF. Okay?
sir. Thank you.
Okay, we are sharing.
Thank you.
Okay, go ahead, Sony.
Okay, sir. Thank you.
Okay.
Good afternoon to everyone. Myself, I'm Sony from Sri Kripa Institute of Pharmaceutical Sciences. Today, my presentation is on kinetic spectrophotometric determination of venlafaxine based on oxidation by alkaline KMNO4. Next slide, please. Contents. Next slide, please. What is kinetic study? Hello. What is kinetic study?
Sony, you are audible. Carry on.
Okay, sir, please. What is kinetic study? Kinetic is the study of rate of chemical reactions. It is used to speed up the reaction that will change under the reaction condition. And this study of kinetics is very important to elucidate the information about mechanism of reaction. And at higher concentration, the reaction will proceed more quickly. And what is kinetic spectrophotometry? Actually, kinetic spectrophotometric method offers an easy and time-consuming sensitive analysis. By using these simple and available reagents, which will be used for routine determination of drug substance. This kinetic spectrophotometric analysis is one of the major interests of analytical industry. Next slide, please. What are the kinetic studies that are important? Why the kinetic studies are important? Why kinetic study are important? There are lots of experiments people perform to work out on how the reaction happens. Chemical kinetics is a measurement how quickly the reaction that will occur. Then if change in condition, it will affect the speed of the reaction. We can learn something about the reaction that will happen, how it will happen. And kinetic studies are important in understanding the reaction. We can also learn something about this, how the reaction will occur. By understanding how chemical reaction takes place, many processes can be improved. Why you have to perform kinetic study? For what purpose? The most important advantage of kinetic study is analysis in the ability to use chemical reaction that will be slow to reach the equilibrium. By using this kinetic method, determination of certain species in a mixture of compounds ought to be possible when species have sufficient difference in the reaction rates. Next slide, please. Aim of the presentation, that is, literature search reveals that UV visible spectrophotometric method is available for estimation of venlafaxine in pharmaceutical formulations, but here no kinetic spectrophotometric method reported for estimation of venlafaxine in pharmaceutical formulation. So the aim of this project is to develop and validate the kinetic spectrophotometric method for estimating venlafaxine in pharmaceutical formulation. The main objectives for these venlafaxine means a selection of solvent and determination of lambda max, optimization of experimental conditions, preparation of standard and stock solutions, kinetic studies, and estimation of drug in formulations and method validation. Scope of these studies are different types of kinetic methods have to be there. That is kinetic method of analysis, fixed concentration method. Next slide, please. Then fixed time method, initial rate method. Now, kinetic method of analysis. An analytical method in which the rate of reaction or a released quantity is measured or else utilized to determine concentration. It is known as kinetic method of analysis. Fixed concentration method. This method is a measure of kinetic analysis in which the period of time required for about the same predetermined change in the concentration of the reactant or product is to be measured. Next, fixed time of method, a mode of measurement in kinetic method of analysis in which the change of parameter related to the concentration of reactant or else product, it is measured over the predetermined time interval. Next, initial rate method. The mode of measurement by which the kinetic method of analysis in which the initial reaction rate to be determined or else used for measurement of concentration. Next slide, please. Literature review. I have literature review. These are the articles I have referred. That is International Journal of Pharmaceutical Science and Research, and World Journal of Pharmaceutical Research, and Journal of Saudi Chemical Society. Next slide, please. Drug profile. I have used the drug venlafaxine, which is soluble, freely soluble in water, and 0.1 normal HCl, isopropyl alcohol, acetonitrile. Structure contains hydroxide group, and one oxygen group and one nitrogen group. The venlafaxine is an antidepressant, and its molecular formula is C12H27NO2. Molecular weight is 277.4, and melting point is 140 degrees C. It is used to treat major depressive disorder, anxiety and panic disorder. Next slide, please. Plan of work, that is literature survey for selection of drugs and selection of specific category of drugs, review literature of selected drugs, procurement of raw material or else reagent, method development and kinetic study, method validation. Next slide, please. Method and materials. Instrument that I've used, that is absorbance measurement was made on Lab India double beam UV visible spectrophotometer with a pair of quad cells, that is 1 centimeter width, and ultrasonicator, then digital weighing balance. Materials, venlafaxine standard purchased from Yarrow products.
Sony,
Yes, sir.
You just make it a little faster, okay?
Okay, sir, sure. Then commercial capsule for Venlor XR, that is a capsule. Then solution of NOH 0.4 normal and analytical grade potassium permanganate I have used. Next slide, please. Procedure for preparation of standard stock solution, that nothing but I have taken 10 microgram of venlafaxine and prepared 1000 microgram per ml. Further stock solution I have prepared 10 microgram, final stock solution 10 microgram per ml. Preparation of reagents, preparation of KMNO4. Standard solution I have taken, that is 0.007 grams of pure KMNO4 and 10 ml of triple distilled water I have used. Then preparation of sodium hydroxide solution, that is 0.4 normal sodium hydroxide. Sodium hydroxide solution is prepared by dissolving 0.4 gram and double distilled water, 10 ml of double distilled water. Next slide, please. Optimization parameters, that is selection of solvent. I have selected water, especially for it is economic free. That's what I have selected. Effect of temperature. Temperature may vary the absorbent results. If temperature varies, the absorbance may increase, and by decreasing the temperature, the absorbance will be decreased. Next slide, please. Effect of time.
Sunny,
Time is
just
an
made the point. Just made the point. No need to go in detail about that. You explain your
okay,
importance
sure,
of
sir.
your work, novelty of your work.
Okay, sure. Effect of time. Time that is exactly 20 minutes. It takes around. Effect of cheminophore concentration. If it is exposed to room temperature, definitely the absorbance will increase. Then effect of NOH concentration. The NOH concentration reaction used the standard is 0.1 or point. Next slide, please. Order of reaction, that is drug and cheminophore and NOH. Next slide, please. These are the results that are obtained, which I was did on my project work. Next slide, please. Calibration graph for every 5 minutes until 45 minutes. Next. Analytical parameters, which I was did. Next slide, please. Reference. Thank you.
Okay. Thank you, Ms. Soni, for
Thank you, sir.
valuable presentation.
Thank
Now
you so
I
much.
request subject expert, Dr. Rand Processor, to evaluate this present.
Yeah, Sony, good presentation.
Reference, sir.
Yeah, and can you explain? Can you compare with the existing methodology, your colorimetric method? Any
I'm
existing
not
method?
getting.
Existing methods, reported methods.
Reported methods, sir.
Yeah.
Of
So this is your work. You have
yes, sir.
performed this work?
My own work, sir.
Yeah. So in the literature survey, I think you should find out some of the other methods, like you mentioned the UV spectroscopic method.
Yes, sir.
So how can you say that your method is superior than existing method? In what way?
Because I have used solvent as water, no, sir. It is an economic free solvent.
Okay. Oh, so what is the solvents you are using for the UV spectroscopic method?
Only I have used water, sir.
No, not the reported method in the UV spectroscopic method.
A reported method is methanol, acetonitrile.
And in terms of accuracy?
Sir, I'm not getting.
In terms
In
of
accuracy.
Sir.
Did you compare the accuracy of your method with the reported method?
No, sir. Actually, accuracy work is going on. Other than accuracy, I did all the work.
That is very important, right?
Yes, sir. I have continued 2-3. I heard. Hello.
Yeah. Yeah, you can continue. Yes, you can continue. We are hearing.
Sir, other than accuracy, I did all the methods, sir, all the parameters.
Okay, okay. Right.
Okay,
Hello,
sir.
Sony.
Sir.
Hello.
Hello.
Any recent applications for kinetics method?
Recent applications.
The multiple component is possible in kinetics method?
No, sir. I had not done it. I have done trials for more than 12 drugs, sir. 13 drugs, I think.
No,
but
I am asking multiple component is possible in kinetics method?
No, sir.
It is possible.
Hello.
Multiple component analysis is possible in the kinetics methods?
Of course, sir.
Okay. Anyway, nice presentation,
sir.
Sony.
Thank you so
Okay,
much.
sir.
Okay, okay.
Okay, thank you. Next participant, Akshay Kumar, Samantha. Akash Kumar Samantha. He was a PG student, NSH Knowledge Campus from Kolkata. Akash Kumar, go for your presentation.
Hello, sir. Hello.
Yes, go ahead.
Good afternoon, sir.
Good afternoon, go ahead, Akash.
Okay, sir. Thank you. My topic is an injectable hydrogel with or without drugs for prevention of epidural scare addition after laminectomy in rats. Okay, so as per introduction, as per the topic, an injectable hydrogel with or without drugs, the prevention of epidural scare addition after the laminectomy in rats is performed. So,
Akash, open your slides.
Akash,
Slides, please.
Open your slides. Open your slide.
can you share my slide?
No.
Okay, yeah. Wait, Akash, wait.
Mandal. Chestunnara?
Can you share my slide? Okay.
Okay, is it okay, Akash?
Okay, sir.
Okay, continue.
Okay, sir. Next slide, sir. Introduction. As per the topic, an injectable hydrogel with or without drugs, the prevention of epidural scare addition after the laminectomy in rats is performed. So before starting, we have to know what is laminectomy. Laminectomy is the operating process that is performed in each and every spinal surgery. In this laminectomy process, we have to remove the laminal part of the vertebra. Okay, then the corresponding spinal surgery is performed. There is an important complication of the post-laminectomy is the formation of epidural scare. Is an defense mechanism of body, okay, that extends towards the nerve roots and causes the compression of the nerve roots and leading to the inflammation and the lower back pain. Okay, to prevent the formation of the epidural scare, we have to inject the hydrogel with or without drugs that can prevent of formation of the epidural scare at the site of the laminectomy. So what is hydrogel? Basically, hydrogel is a network of polymer chain that are hydrophilic, sometimes found as a colloidal gel in which water in the depression medium. So at the side, okay, there is the hydrogel is PLGA, PEG, and PLGA, tribocopolymer into which we may have added the DX or may not. Okay, DX means dexamethasone is a steroid hormone, is anti-inflammatory. It prevents the inflammation of the site of the laminectomy. Next, materials, chemicals and animals. Chemicals, PEG, stannous, octocronal, just others. Next slide. Okay, method. A few methods are here. First, in vitro degradation. In vitro degradation study of the hydrogel performed in aqueous solution. Okay, the aqueous solution is stored into the vials and it is stored at the temperature of 4 degrees Celsius for 12 hours. Then the sample is incubated with constant shaking for the formation of hydrogel, and then it is lyophilized until constant weight. For the analysis of the sample measurement of GPC, gel performance chromatography, weight and weighting were performed. The pH was also recorded with pH meter. Next, in vivo degradation. In vivo degradation is performed inside the body of an animal, and the animal is Sprague-Dawley rats. Okay, and aqueous solution of hydrogel was made, that is the 25% by weight in NS, normal saline, which was sterilized. 11 rats was anesthetized and hydrogel was introduced via SC routes, subcutaneous routes. In after scheduled time, the animal was sacrificed and remaining gels that prevent inside the body of an animal is observed for the remaining about the hydrogel. Next, in vitro release of DEX. In the in vitro release of DEX, we have to determine the release pattern at the route of release, the dexamethasone from the hydrogel system. The drug content in the release medium was determined the HPLC and UV visual spectrum. Next slide. Next slide, sir. No. Previous slide, surgical procedure here. Yes, okay. This is surgical procedure. At first, the anesthesia given to the rats, then a longitudinal midline section was performed over the L2 to L4 level. Then the paravertebral muscle was defected. Then at the L3 level, a complete laminectomy was performed and dura mater was completely intact. Okay. Now the rats are divided into 5 groups. The control groups, for the control groups, no hydrogel was given. Next, hydrogel group, only hydrogel was given. And hydrogel plus drug treated group, there are 3 concentration of drug hydrogel that was given to the rat. In the concentration are 1 mg per ml, 2 mg per ml, 4 mg per ml. After the introduction of hydrogel, the muscle and skin was used. The rats were referred into their cage and they were carefully. Finally, they were sacrificed by in the peritoneal injection of the phenobarbital at 4 weeks post-operation. Next slide. Next slide, sir. Okay, observation and result. First, 2 observations are there, clinical and macroscopic. First, clinical. Neurological function and the development of wound healing was observed after 2 hours of the post-operation, and the subsequent 3 points was sorted until they were euthanized 4 weeks later. Number 1 observation, the posture of the effective hind limb. The deformity of the hind limb was graded according to the following: 0, 1, 2, 3. 0 complete deformation, 1 important deformation, 2 slight deformation, and 3 normal. And number 2, the observation is the force and use the hind limb. 0, 1, 2, 3, 4, 5. 0 complete paralysis, and 1 the hind limb muscles can be contracted but limb cannot mobilized, and 2 hind limb can be moved horizontally but cannot support the body weight, and 3 hind limb can support the body weight but some difficulty. Okay, next 4 force and use the hind limb were nearly normal, and 5 force and use the hind limb are normal, but animal whose neural roots were injured in operation were finally excluded in the following study. Okay, next. Next, macroscopic observation of epidural scare addition. Okay, now the macroscopic observation of epidural scare addition, it was performed after the animal was euthanized. From each group, animal was selected and they were euthanized 4 weeks after the operation, and histological parameters and histological observation was performed on the site of laminectomy. The formation of epidural scare on the site of laminectomy was graded according to the following parameter. Grade 1, 0, 1, 2, 3, grade 0, dura mater is free of scare tissue, and grade 1, only thin fibrous bands are observed between the scare tissue and dura mater. Next, grade 2, continuous adherence is observed and less than 2/3 of the laminative effect. And grade 3, scare tissue adherence is large, affecting more than 2/3 of the laminectomy defect, or the adherence extend to the nerve roots. Okay, next slide. Okay, result. In vitro degradation of the PLGA, PEG, and PLGA hydrogen. Okay, of the hydrogel was performed in the aqueous solution of 25% by weight after the incubation, and it was seen that the volume has been increased over 100% after the incubation in the PBC at 37 degrees Celsius for 5 days. Afterwards, the swelling rate of hydrogel is slightly slowed down. We can clearly see these phenomena in this graph. Okay, first graph, we can see the degradation time, gel volume. Okay, the degradation time is increased, the gel volume also increased, but after some time the slope tends to decrease. Next graph, the weight of the hydrogel decreased to about 50% after 5 weeks of degradation, and this properly we can also observe by this graph. And x-axis belongs to the degradation time, y-axis belongs to the gelling weight. At degradation time increase and gelling weight decrease, so it is very effective in vitro degradation. Third graph, a slow increase in the retention time has also been observed in the in vitro degradation study of the hydrogel. We can clearly see a slow increase in retention time. As far as at retention time increase, the increase in the retention time tends to decrease. At a particular time period, the retention time is back at maximum level. Fourth graph, as per the graph, we can see that y-axis at the left side belongs to the M peak, and y-axis at the right hand side is corresponded to the molecular weight distribution, and the x-axis correspond to the degradation time. With increase in retention time, the molecular weight peak in decreasing, but increase in the retention time, the molecular weight distribution is increasing. Fifth graph, y-axis and x-axis degradation time, increase in degradation time, we can see slightly fluctuation in pH of the medium. Next, in vitro release, the property of the previous slide.
Akash, you can see the figures and explain. No need to read all the lines. You can see the figures and explain. No need to read all the lines. Only explain the figures. Make it fast.
Hello. Okay. Next, in fiber degradation, this picture first shows the remaining amount of the hydrogel that was present under the screen. Second this shows the different side of hydrogel and different time of intervals. So as the degradation time increase, the in fiber degradation of the hydrogel in occurring, and that's why the size of the hydrogel increasing. As the size of the hydrogel is decreasing, we can conclude the next graph, that is the degradation time versus gel remaining. This is gel remaining weight percentage. As degradation time increase, the weight of the hydrogel remaining inside the body, gravity is decreasing. And lastly, this is the GPC profile with degradation source. Next slide. Next, histological. This first table. This table is the histological evaluation of the different groups, and groups are divided into 4 different grades according to their histological parameter. For the graph A, for the control group, we can see all animals are fallen under the grade 3. For group B, for this group, we can see 4 animals were under grade 1 and 2 animals under grade 2. And group C, 1 animals under grade 2 and 5 animals under grade 3. And group D, 3 animals under grade 1 and 3 animals under grade 2. And group E, 1 animals under grade 1. Okay, next. Okay, conclusion. In the article of the prevention of epidural scare laminectomy, it has been clearly observed that the application of hydrogel, that is PHA, PEG, PNDA, tribaclopride, has shown as an effectiveness towards preventing epidural scare, and inflammatory steroid hormone has also been incorporated in the hydrogel system to prevent the inflammation, but it shows a complicated result. There are 3 concentrations, 1 mg, 2 mg, and 4 mg per ml, but only 2 mg per ml concentration of the steroid hormone shows maximum effectiveness against epidural scare and to prevent the inflammation. Okay, thank you.
Okay. Thank you, Mr. Akash Kumar. Now I request subject experts to evaluate this presentation. Please ask, sir.
Mr. Akash,
Sir.
yeah, this is your work.
No, just research article and I'm a review.
It's a review article, right?
Okay.
Exactly.
So, so actually, I would like to ask some questions on some in vitro and in vivo analytical related things. Since it's a review article, I don't actually. What kind of column you're used for HPLC? Do you get any information from the review? Hello?
No.
No?
Hello.
Okay, right. Carry on.
Hello, Akash.
Yes, sir.
Hydrogel is a what kind of dressing?
Yes, sir. Please repeat.
Hydrogel is a what kind of dressing?
Wound healing that.
Hello, pardon? Gel based or wound?
Wound healing. Gel based wound
Okay.
healing.
Okay. It is hydrophilic or hydrophobic nature? Hydrogel. Hydrophobic. The gel, most of the gel is a water, I think, in the hydrophilic polymer, I think so. Ranjit Prasad, sir? I think it's hydrophilic nature, I think. Okay. Anyway, nice presentation.
Thank you, sir.
Thank you, sir, for evaluation. Next, I invite Preetham Deka. He was a research scholar from Assam. Preetham Deka, go for presentation.
Hello. Am I audible?
Yes.
Yes.
Okay. Good afternoon, everyone. So today I am going to discuss my research topic on in vitro antibacterial
Slide,
activity
please.
of
Slides.
Pritam, do you have slides with you, or you'll play from here?
Sir, I have submitted.
That. Okay. Wait.
Just wait.
Is it the slide, Peter? Is it okay, correct?
Yes,
Go
sir.
ahead.
Yes, sir.
Go ahead. We
Thank
have
you, sir.
less time. Go ahead.
Yes, sir. So I am today going to discuss my research topic on in vitro antibacterial activity of substituted chalcone derivatives. This work was done in my bachelor project by green synthesis procedure. Now I am doing MPharm. Project was done in minimum resources and minimum time. I would like to thank Pharma Analytica 2020 for giving me this opportunity. Next slide. Hello. Hello. Am I audible? Hello.
Yeah, you are audible.
Coming to the introduction part, chalcone is a aromatic ketone and anion that forms central core of variety of important biological compounds, which are known as collectively as chalconoids. Chalconoids are also known as benzyl acetophenone. The presence of anion in chalcone confers biological activity upon it, like antibacterial activity, anti-inflammatory, antioxidant, antifungal, anti-tuberculosis, and many more. The presence of a reactive alpha and beta unsaturated keto function in chalcone was found to be responsible for their antimicrobial activity. Next slide, sir. Now, aim and objective. From various literature survey, it was observed that chalcone have antibacterial activities. So our aim is synthesis of chalcone derivatives by casein condensation and investigate and evaluate their antibacterial activity. Next slide, sir. Material. These are the material I have used. We took aldehyde as a compound because it is easily found in our university lab. And these are the instruments which I use. Next slide, sir. Hello. This is the table of compound along with their code, which we consider A1, A2, A3, A4, A5, A6, A7, and standard SS. Next slide, sir. Hello. Next slide.
Just wait.
Sir. Yes, sir. Previous one, sir.
जजमेंट के पहले मैं आपको एक थोड़ी सी पनिशमेंट देना चाहता हूँ।
Hello, Peter. Are you able to see the slides?
Sir, previous one. Yes, sir. I can see.
Previous slide. Previous.
Yes. Thank you. These are the method by using synthetic method. We use Claisen-Smith concentration. The most convenient method is equimolar quantities of aryl methyl ketone with aryl aldehyde in presence of alcoholic alkali. Procedure: the synthesis of chalcone was carried out by this concentration process of commercially available acetophenone and benzaldehyde in presence of sodium hydroxide as a base in ethanol. And the reaction was initiated by removal of a proton from a carbon of acetophenone to form a resonance stabilized enolate ion by the base. This was followed by the nucleophilic enolate attacks on electrophilic carbonyl carbon of benzaldehyde, resulting in a new carbon bond formation. This reaction joined a carbon of acetophenone to carbonyl carbon of benzaldehyde to form intermediate. The final step of this reaction was protonation and deprotonation by hydroxide ion to form an alpha beta unsaturated ketone, TLC. It is used to find out the purity of the compound by taking suitable solvent as mobile phase and silica gel as stationary phase. Infrared spectrophotometry. The infrared photophotometry spectrum is unique for any given chemical compound, with exception of optimal isomer, which have identical spectra. It is possible to measure quantitatively the individual component of a mixture of known qualitative composition without prior separation. Antimicrobial activity. Antimicrobial activity, we use Dix diffusion method. Dix diffusion method, Salcon derivatives procedure was Salcon derivatives were dissolved in distilled water, that 100 mg and 1000 mg Salcon derivatives were taken in the beaker, and 1 ml distilled water was millipoured. Then nitron agar was prepared in sterile conical flask, autoclaved for 30 minutes, then poured into sterile petri plates, and the plate were taken inoculated with sensitive bacteria, which was Staphylococcus aureus. The two diameter dish was made and dipped in respective Salcon derivative solution, 100 and 1000 micrometer per millimeter, and placed on the culture media. Then the plates were inoculated at 37 degrees Celsius for 24 hours. After that, result is observed and zone of inhibition is measured. Then result and discussion. Result and discussion. So result and discussion. Yes, sir. In first table, from all the synthesized compound, A1 is having the highest percentage yield, and A1 plus A3 have lowest percentage yield. Melting point of synthesized compound is given in that table, and the entire compound shows the sharp melting point. And the second table, all the compound are taking, and first for initial, all compounds are grinding singly, and then very small amount of solvent and check their melting point in melting point apparatus. For final, mix two compound and grinding that mixer, and then add small amount of solvent and check their melting point in melting point apparatus. Next slide, sir. Okay. For TLC, we measure the RF value. RF value was distance traveled by solid by distance traveled by solvent, and solvent was taken methanol by 1:4:5, ethyl acetate, methanol, and chloroform. Next slide, sir.
Hello, Pritam, continue.
Sir, next slide. Yes, sir. Spectral analysis. For spectral analysis, we use FTIR because it will show compound graph. From that, we will get peak picking of compound. After getting peak picking of compound, it easy to find out the functional group and adsorbance of that compound. After doing FTIR, most of the graph of compound get broad peak, and broad peak always define OH group. Somewhere we will get carbon group, which most probably single peak. NH group, that is half peak. These are the picture. You can see broad peak is there. Next lecture. Okay. Zone of of inhibition. The zone of inhibition is a circular area around the spot of stand in which the bacteria do not grow. After processing antibacterial activity, calculate the diameter from standard to area A1 and A2 compound level. Like this, all the table are calculated. Next slide. Okay. Conclusion. Number A, inhibit the bacteria of Staphylococcus aureus. B, inhibit the activity of that bacteria. C, shows reasonable activity of that bacteria. A plus B, inhibit growth of bacteria and prevent activity. And B plus C, show reasonable activity against bacteria. So it can be used to treat infection caused bacteria such as urinary tract infection, diarrhea, meningitis, sepsis, reference. Next slide, sir. Reference. These are the reference and many more which I followed.
Okay.
Thank you.
Okay, thank you, Preetham, for a very good presentation. Now I request subject experts to evaluate this presentation.
Yeah, Preetham,
Yes, sir. I can.
I have so many questions. I think you did not reveal your structure of the molecules you synthesized.
Exactly.
Okay, in the entire presentation. So it is a wonder. Without showing the molecular structure, you completed your presentation. Okay, and okay, and I have my own doubt when you look at when I look at the physicochemical characteristics, there is a melting points. So some of your molecules are having melting point 40 degrees and some of them are 110, 120. With the same nucleus, chalcones are showing the different molecular weights. Okay, I don't think your molecules are having that much difference, the same nucleus. Okay, and also you mentioned that during the biological property, you grinded your molecules. Your molecular melting point itself is a 40. It is very close to room temperature. So when you're handling itself, it will be like it is in the melting stage. Okay. I think this is your senior's work. Preetham,
answer. Preetham, unmute yourself.
hello.
I think he left.
Okay, good.
Preetham.
Hello.
Hello, Preetham.
Hello.
Preetham came. Okay. Give the answer. What Sir is asking.
Sir, I have done this work in my BPharm level, so I am just evaluate and see this research topic on research.
Okay. So maybe at the B form level work, maybe
Yes,
the reason is your molecule is not you're not able to synthesize the molecules properly.
Yes.
So you're characterized only by FTIR. Okay, you won't get anything information characterization data only with FTIR. Okay, and one more thing I would like to suggest you for the biological property, as that is antibacterial activity. Should not compare molecules like chalcones with broad spectrum antibiotics like amoxicillin and ampicillin. You did. Okay, your selected standard molecules are the amoxicillin and ampicillin, right?
Yes, sir.
Should not select ampicillin and amoxicillin. They are broad spectrum. We cannot expect your chalcones are the simple antibacterial properties we can expect. Instead of that, we can select some other antibacterial molecules like quinolines or some other. Okay. Anyway, good attempt. Okay. And
thank you, sir. Thank you.
Thank you, sirs, for your evaluation.
Thank you.
Okay. Next, I invite Mr. Royal Debnath. He was from Sri Kuba Institute of Pharmacosciences, Siddipet.
Audible, sir?
Royal Devnath, are you ready for the presentation?
Yes, sir. Hello.
Did you hear me? Royal Devnath,
Yes, sir. Hello. Can I
are you
audible,
ready
sir?
for presentation?
Yes, sir.
Yes.
Do you share or we will share from here?
Yes, sir. Sure, sir.
You are going to share, no?
Okay.
Okay, share yourself.
Sir, you share. I think it's not there, sir.
Okay. Sir, welcome Dr. T.G.K. Murthy, sir, Babatla College of Pharmacy Principal. Sir, welcome, sir.
Good evening to all of you.
Sir, good evening, sir. Very happy to hear, sir.
Okay, thank you.
Sir, thanks for presence here, sir.
Okay.
So, Royal Devnath, I have shared your PPT. You just go on.
Okay, sir. Thank you. Sir, good afternoon, sir. Sir, I'm audible, sir. Hello.
Yes, you can go for it.
Okay, sir. Good afternoon, sir. Myself, Royal Devnath. I am from Sirkuba Institute of Pharmaceutical Science. Today, I'm give my presentation work is development and validation of new visible spectrophotometric method for the estimation of glucose in bulk and pharmaceutical doses. I thanks to Pharma Analytical in 2020 to give me an opportunity. Next slide, sir. Next slide, sir. Hello, sir. Next slide.
Wait few seconds, Devnath.
Okay, sir.
Yeah, Royal Devnath, you can continue.
Yes, sir. Introduction. Analysis is an important component, sir, the foundation development of any drug because that our main concern is development validation of visual spectrophotometric method per ICH guidelines. Thus the present study was developed and related simple, sensitive, accurate, and reproducible visual spectrophotometric methods for the
Royal Devnath, 1 minute. You just reduce the introduction part. You go for the result discussion and explanation and what is the novelty of your work.
Okay.
You explain that one.
Okay, sir. Okay, sir. Next one, sir. So this aim is, sir, presentation main aim is to develop visible spectrophotometric methods for estimation of bug levels in API and pharmaceutical formulation. These are the literature review of visible spectrophotometry. And I got, sir, only one literature review in that use lambda 582 nanometer and solvent use methanol. Yes, sir. Next. The drug profile, sir. The drug name is Boglibos, category is anti-diabetic, It is used to treatment type 2 diabetes mellitus. This drug was soluble in water, methanol, 0.1 normal sodium hydroxide, and 0.1 normal hydrochloric
Say
acid.
louder,
The
Royal
drug was
Devnath. Say
insoluble.
louder.
Yes, sir. Okay, sir. This is insoluble in sir chloroform, isopropyl alcohol. These are the structure of Boglibus. Next slide, sir. At the methodology, sir, the instrument are used to use, sir, that Helicos spectrotrace SL244 and Lab India UV spectrophotometer with a pair of matched quad cells 1 centimeter width. That ultrasonic are used for the drug and sample solution for the sonic. The digital weighing balance used for the weighing. That materials are Boglibus standard gifted from the pharmaceutical industries. Then commercial tablet from Volvo containing 0.3 mg of Boglibus was purchased from the local medical store. That solvent used hydrochloric is a analytical reagent grade, and that analytical grade reagent like potassium ferricyanide and ferric chloride used for the visible purpose. That method was the first selection of suitable solvent, then preparation of standard Tox solution, then detection of lambda max, then preparation of test solution, then estimation of Boglibus. In that ICH guide, ICH parameter like linearity, assay, accuracy, limit of detection, limit of quantitation, ruggedness, and robustness. Next one, sir. That the preparation of solvent like 0.1 normal of hydrochloric acid, the concentration of 8.5 ml of hydrochloric acid dissolved with 1000 ml distilled water from 1000 microgram per ml, and preparation of reagent like 0.5% weight by volume potassium ferricyanide solution. It is prepared dissolving 0.5 gram of potassium ferricyanide dissolved in a distilled water to form a 0.5% weight by volume. This is the preparation of standard and test solution. How to prepare standard solution? First, weight accurately sir 10 mg of drug and dissolve in a 10 ml of volumetric flux. Make up with 0.1 normal of HCl. Then in that it is from 1000 microgram per ml. Then in a take pipette out 1 ml and make up with 0.1 normal HCl it from 100 microgram per ml. In that pipette out 2 ml, 2.5, 3, 3.54 and make up with 10 ml of volumetric flux with 0.1 normal hydrochloric acid, add 1 ml of potassium ferricyanide and 0.1 ml of ferric chloride to from 20, 25, 30, 35 and 40 microgram per ml and measure the absorbance. In the test solutions are accurately 5 tablet are weighed, then average weight are calculated, then crush the tablet in mortar and pestle, then powder equivalent to 1.5 mg of Boglibus is transferred into a 10 ml of volumetric flask. Then take little quantity of 0.1 hydrochloric acid, sonicate for few minutes, then filter the solution and pipette out 2 ml, then add 1 ml of 0.5% of potassium ferricyanide, then 0.1 ml of ferric chloride and make up with 0.1 normal hydrochloric acid. Then is from 30 microgram per ml, then measure the absorbance. These are the resultant discussions, sir. This lambda max was found 775 nanometer, and the solvent used are 0.1 normal hydrochloric acid. These are the sir visible spectra. We use 20 to 40 microgram per ml. Next slide, sir. Sir, assay. Assay is used to perform to determine the presence of substance and the amount of this substance. Assay is an investigative analytic procedure in lab medicine. And you concern that percentage purity should be 95 to 105% in between. Then accuracy. Accuracy means measurement of nearness of the true value to the measured value. That standard deviation should be greater than less than 2. Then limit of detection and limit of quantitation. That LOD, lowest concentration of the instrument is able to detect but not quantify. In LOQ, that lowest concentration is detect but and quantify. Next one, sir. These are the precision value. Precision is defined as the measure of closeness of concurrent reading. Precisions are two types, sir. Repeatability, precision, and reproducibility. Repeatability means sir morning, evening, and reproducibility means day 1, day 2, day 3, day 4. Their standard deviation should be less than 2. Then sir, ruggedness. Ruggedness is a measure of reproducibility of the test result under normal. Then robustness. The robustness. These are the summary validation of parameters for the proposed that solvent used, linearity range, correlation coefficient, acceptance criteria, sir. Then array 90 to 105%, then limit of detection, then limit of quantification. That's the sum I'm using.
Okay, thank you. Okay, thank you, Royal Devnath.
Thank you, sir.
Now I request sub-experts to evaluate this presentation.
Good presentation, and this is your work?
Yes,
As a
sir.
review.
No, sir. My M.Pharm.
Your M.Pharm work. Okay. What is the lambda max?
775 nanometers, sir.
How much?
775.
But you have shown that somewhere it is a 550.
No, sir.
In the literature, you are shown. You only shown. In
This
your
is
presentation.
literature reviews, sir. Another literature, not my.
Okay. Which one is correct then?
My one, sir.
Yours
This
is correct.
This one, sir, solvent use methanol. I am using, sir, 0.1 normal hydrochloric acid.
Okay.
My lambda max was, sir, 775 nanometers.
So what is the absorbance you have observed during at the 770?
Yes,
How
sir.
much absorbance?
Hello, sir.
Any idea?
Absorbance, sir.
Yeah. Means the shape of the peak. Okay, there is a slight difference around 550 also and like 750, 775.
In 775, sir, more absorbance came, sir.
Okay, okay. Then how your method is superior than reported method?
Yeah, using, sir, methanol, sir. I'm using in low cost solvent like 0.1 normal hydrochloric acid.
Okay, what about other reagents?
Other reagents are ferric chloride, they're coloring reagents. Ferric chloride and potassium ferricyanide.
Okay, then what is your justification? Your method is in what way it is superior?
Cost of effective, sir.
Okay, okay, carry on. Good.
Sir, Dr. M. Srinivas Reddy, sir, ask any questions for this presentation.
Yeah, same questions are asking, sir. No problem. Carry on.
Sir, next, Dr. T.G.K. Murthy, sir, ask any.
Chalo. Okay. With respect to analytical work, previously I also witnessed Sony's work. In both the cases, my point is, as you observed spectrophotometric method, visual spectrophotometric method, there will be development of some chemical reaction. So what is the chemical reaction between your drug and ferric chloride, and what is the color observed? Because in case of visible spectrophotometric method, there should be a chemical reaction between the drug and the reagent used. So what is the reaction taking place between the drug and ferric chloride? Am I audible?
Hello, sir.
Yes, sir. Yes, sir.
Yes, sir, it's clear.
Okay. Because in earlier Sony case also, there was no explanation about the mechanism of chemical reaction. That is the one of the required parameter between the drug and reagent. and what is the name of the product or complex formed, what is the color observed, and because of that, wavelength should be selected. As in case of visual spectrophotometry, we are having VIBGYOR. So with respect to each color, there will be a particular range. So what is the color observed, and accordingly, the wavelength selected.
Okay, sir, I will do, sir.
Whether Royal Devnath, whether you are having answer or not.
Sir, this one.
Devna, respond to sir. Say yes or no.
Sir, I will do, sir. I am not perfect.
Means what is the color you noticed?
Blue color, sir.
Blue color.
Yes, sir.
What is the color of ferric chloride?
Ferric chloride, sir. Crystal red color, sir.
Because you prepared ferric chloride reagent.
Yes, sir.
So what is the color of that ferric chloride? Whether it is having color or it is free from color?
Color, sir.
Then
No,
what is
sir,
the color?
Sir. Red, sir. Crystal red.
Okay.
Okay, sir. Thank you, sir. Okay. Thank you, Mr. Rayal Devnath, for your presentation. Next participation, may I request Mr. Arif Pasha, Associate Professor, Vageshwari College of Pharmacy, Telangana.
Hello.
Arif Pasha.
Hello. Hello.
Arif Pasha, go for presentation.
Yeah, good afternoon. Is it visible?
Yes, it's very clear. Go for presentation.
Okay, good afternoon. So, one of my research work in my PhD, I have synthesized avant-garde synthesis of 3,4 substituted pyrols. Okay, from by doing molecular modeling in silico, ADMET, along with some sort of pharmacological activities. So this is my graphical abstract. I had synthesized a sequential addition reaction in which synthesis of chalcones will be carried out, then followed with Tosmic. So formation of pyrrole derivatives. To that pyrrole derivatives, screening has been done on in vivo studies. In that, I have isolated one molecule which is having anti-ulcer activity and as well as one as anti-inflammatory activity. Both these molecules, whatever the molecules that are having some sort of anti-inflammatory activity, it is showing some sort of ulceration in the stomach. So I have concentrated on this one by doing some predictive studies like drug likeliness and ADMET studies. So this is my abstract. So for in vivo studies, I have got an anti-inflammatory activities of inhibition of 80.65 at a graded doses. I had increased the doses by taking 1/10th and 1/20th part of the acute toxicity test, then followed with anti-ulcer activity with 77.5 of one pyrrole molecules at 200 mg per kg. Then molecular modeling has been carried out on 3 proteins. One is COX-1 and one is COX-2 and another is H plus K plus ATPase enzyme, that is anti-ulcer, and followed with toxicity prediction and drug likeliness studies by Osiris Property Explorer and Admit SAR prediction tools. Out of this, I got two molecules that I have already told you, that is PY1 and PY5. PY5 having good anti-inflammatory activity and as well as PY1 having good anti-ulcer activity. So this is my schematic part. Like simple condensation reaction of aldol. with the help of formation of chalcones by aldehydes and ketones, then followed with plasmic for getting your work pyrols 3+2 cycloaddition reaction. So these are the molecules that I have done. Around 12 molecules I have synthesized. Uh, so continuation to that one. So what are the procedure I have taken part? So it's a simple schematic procedure for which normal ethanol will be taken in the part with the help of a small base lithium hydroxide in millimolar concentration a little bit. Stirring has been carried out, then followed with stirring. Hello? Hello?
Yes, sorry, continue.
Yeah. Stirring has been carried out for 2 to 3 minutes, then followed with aldehydes addition. So till the reaction, this is a sequential addition reaction. So we have to monitor the reaction with the help of some solvent systems. So I have taken hexane and ethyl acetate as a solvent system, for which I have got a separated spots, then followed whatever the formation of chalcones that has been carried out, then followed with the sequential addition reaction again by adding the base and as well as a plasmic molecule till the precipitate of the formation has been done, then followed with washing with water and cold ethanol successively. Why I am washing with cold ethanol means, so whatever the molecules that have been formed, like any unreacted compounds like aldehydes and ketones, it will be solubilized in cold ethanol only. So any unwanted or undesired product that has been got, that has been eliminated here itself. So these are my one of I couldn't able to show you all the 12 slides, 12 IR NMR mass slides. So I am showing one of the compounds of one IR of one compound which I have got an anti-inflammatory activity PY5, that is one. Then followed with my NMR studies of PY1 compound. So I have got one molecule NH, formation of NH ring here. So we can see 1.133. So pyrrole molecules I am getting here. Then followed with, so I have, based upon the literature survey, I have got that some molecules with pyrrole I can see anti-inflammatory activity, anti-ulcer activity. So here, whatever the 12 compounds I have synthesized, I have went for in vitro anti-inflammatory activity by taking diclofenac as a standard form as in bovine serum albumin denaturation methods. So out of which I have got around PY1 PY1 molecule and as well as PY5 molecules. Along with PY5, I have got PY12. These three molecules I have isolated for better activity. So I can go through the in vitro studies which I have got a good in vivo activities. I have forwarded with the in vivo studies. So in vivo in vivo activities I have performed on these three molecules by some graded doses. Already I have told you, based upon 1/10th part and 1/20th part of acute toxicity studies. So I had went PY1 compound, PY5 and PY12 compound. I'm taking the same similar diclofenac what I have taken in vitro studies. So in that I have seen some molecules like PY1, whatever the in vitro studies, it is showing good, not here it is showing by carrageenan induced rat poedema method. So you can see here my results. PY1 I am getting up to 3 hours. Percentage change in POWAL around we are getting 29% only. So if I increase the dose for 200 mg, not much mark upgraded here. Any graded doses, if you are increasing, also we are not seeing any sort of activity till third hour. So when you see a molecule as PY5 and in graded doses 100 and 200, I'm seeing some sort of activity here. So 54 I'm seeing here percentage change in volume, and as well as 200 for 200 I'm getting 80 percentage. So similarly, PY12 also we are not seeing much amount of increase when compared to the diclofenac standard one. So whatever the molecules I have done for in vivo studies anti-inflammatory, again I have went for in vivo anti-ulcer activity molecules by taking standard omeprazole as a drug. So the same thing I am seeing here. So here different results I have got that PY5 whatever I had shown in I have got in vivo anti-inflammatory activity. I am not seeing any sort of activities in PY5 here. So I can get a PY1 as good anti-ulcer activity compared to PY5. So PY12 is acting as a moderate form. So we can see here in vivo activities PY12 here 35 and 48 and PY12 in anti-ulcer activity 56 and 68. So whatever the molecule I am getting here of highest dose as PY1 200, we are getting 77.5% inhibition of ulceration we are seeing. So these are my diagrams which I have got for by doing anti-ulcer activities. This is the control group and this is the treated group omeprazole. 40 mg itself I am taking here. Then followed with PY1 and then it followed with PY5 and PY12 of 200 mg doses I have kept here. So I have seen clearly that PY1 is showing good activity, so mild ulcerations are not visible here, showing normal mucosa here. So for that reason, so again I had underwent some sort of docking studies for which docking results are being here. So two molecules, whatever I got here, PY1 and PY5, I had docked for COX1 inhibitor, sorry. I had docked for COX1 inhibitor, I docked for COX2 inhibitor of the two molecules, and as well as simultaneously I docked with H plus K plus ATPase enzyme molecule proteins. So I have got the different results here. So when you see a molecule, so binding energy is lesser when compared to PY1. So this is used for anti-ulcer activity in my research, but actually here binding energy is lesser to some extent. So this can't be estimated. When binding energy is less, you can't estimate a drug to be a less potent. So I had seen the molecule for here number of amino acids, bonding is there. So two bondings with the two amino acids bonds are there. Here also in COX2 also two bondings are there. When you see PY1 for anti-ulcer, we are seeing two amino acids, it is bond. The conventional bonding, hydrogen bonding is taking place more compared to PY5. So concluding this one as taking as a bonding interaction will be more for PY1. So I had chosen this one for anti-ulcer activity and PY5 for anti-inflammatory activity. When you see for diclofenac, so here itself also I have taken diclofenac, which is bound to 3N8Y protein and as well as 1PXS protein. So when you see a molecule with anti-ulcer activity, so it is having lesser minus 5.34, which is around to that one the same binding energy which is having for my compounds also. So this one I have taken SCH28080. This is one compound which is bound to some protein which is having H plus K plus ATPase enzyme. So I had compared this compound also, which is having the same binding energy as compared to this one. So then docking results. So these are my docking results which I have got, whatever the results that I have placed here. These are my docking results. So conventional hydrogen bonding we can see here in a green mark. Okay. Followed with pi stacked system is there, pi alkyl system is there. So here also the same thing, py5. This is py1 docker molecule and this is py5 docker molecule. So whatever the docker systems I have done, that I have done for whatever the molecules I have seen here. So all molecules I have run with some sort of drug scores and as well as toxicity risk why my molecules has or not. So overall findings, I had seen that my drug score when compared to PY1, PY5, drug score is good compared to diclofenac and omeprazole when you see the drug score. And as well as when you see the drug likeliness also, PY1 is having good drug likeliness compared to PY5. And here you see, even though it is in minus form, drug likeliness is low. So whatever the concentration, whatever the drug likeliness, higher the number, higher the chances of getting a drug into clinical trials. So my drug score is also good compared to standard forms like diclofenac and omeprazole. So I'm getting 0.9 and 0.84. So when you see a molecule at diclofenac, so I had searched in Osage Property Explorer that some sort of reproductive effect exists in diclofenac. Whereas if we see in the molecules what I have synthesized, PY1 and PY5, I got negative results when you see the toxicity risk. Then this toxicity risk I had went for again ADMET properties. So whatever the drug that I have is of any carcinogenicity is there, any absorption is there or not in that molecule. So whether it is reaching blood-brain barrier. So when you see a molecule, human intestinal absorption, my drugs are getting absorbed, means it is reaching, it may reach its target site because of its absorption is 0.98 percentage. When you see a diclofenac as 0.96. So omeprazole 0.99, so little bit near to about 0.98. Okay. So most of the drugs, whatever the drugs I have chosen here, it is having good intestinal absorption and it is reaching its blood-brain barrier as 0.97. When you see a PY1 molecule for anti-ulcer activity, it is 0.95. Means some sort of good uh, absorption we can see here. So when you see the carcinogenicity of the molecules, none of the molecule, whatever the synthesized is non-carcinogen. And when you see the LE50 values in rat, we can see that around 2 kgs per mole molecule. If we take also, nothing will happen. So no toxicity, lethal dose is around 2. So when you see compared to omeprazole, 1.7 here. So my drug is somewhat safer one compared to. So I had concluded with this one a series of neoteric substituted pyrols synthesized, and my two molecules what I have selected for anti-inflammatory and anti-ulcer activity. It is showing some sort of moderate activity compared to in dose dependent manner, and if any further modifications can be made for better activity, and we can see more than less toxic effects can be seen here. So with this I will conclude. Thank you for the PHARMA ANALYTICA team.
Okay. Okay. Thank you, Arif, for your very good presentation. Next, I invite experts to evaluate this presentation.
Hello, Arif.
Arif,
Yes, sir. Sir, good afternoon.
Arif.
Arif,
Good afternoon, sir. Good afternoon.
yeah, Arif, a very good presentation. On what basis you are selected and during the process of drug design, you are selected 3 proteins, right?
Yes, sir.
So on what basis you are these proteins
Sir,
from
on
PDB?
what? Yes, sir. Yeah. On what basis means whatever the proteins I have selected, like for example, 3N8Y and 1PXS and 2XZD. So
Yeah.
3N8Y, it is docked with, already it is docked with diclofenac. So I'm comparing a molecule with diclofenac. So whatever the OX2 is also there, 1PXX, it is also docked with diclofenac itself. So when comparing, we are
Yeah,
comparing
yeah.
with diclofenac.
So that is my major question. First of
Yes,
all,
yes.
why you are selected diclofenac? Any relation between like in terms of mechanism or in terms of
Drug
structural?
structure, drug structure, drug
No,
structure
no,
not
sir.
related to diclofenac.
Not related to diclofenac, but mm, common drug, no, sir. So any less sort of lot of older drug which is having some sort of safer forms.
Converts back to empty.
Okay, the omeprazole also.
Omeprazole also oldest drug, no, sir, with a safer and with a lesser side effects it is there. So when you compare, we can compare with older drugs. When you compare with older drugs, whatever the new drugs are there, we have to show somewhat good activity or good. If it is good active potent compound, we can get good molecule. We can sort it out when there are older drugs to conform
But
into any.
I believe in place of the diclofenac, if you are able to select any other selective COX-1 inhibitors.
Yeah, sir. We can select, but actually, when we are selecting diclofenac, when I choose the diclofenac, but molecules or proteins are there which is already bound to diclofenac, which is having good sort of. When it is used for OTC counter drugs, why can't a molecule will be synthesized safer than that one like that?
Okay. Okay, right. Good. Thank you.
Hello, Arif. Hello.
Sir. I can hear.
Very nice presentation, Arif.
Thank you, sir.
Hello.
Sir, tell me.
Yeah. In the while preparation of your compound,
Yes, sir.
what is the importance of lithium hydroxide?
It's a base, sir. So any molecule, if you want to do any aldol condensation reaction,
Okay.
so obviously base is required.
In that your preparation, how to handle the lithium hydroxide? It is a hygroscopic nature.
Yeah, sir. Actually, it's a hygroscopic nature. So
Yeah.
molecule itself, it is getting in a crystal form with the help of water molecule. So molecule, whatever I am getting, lithium hydroxide monohydrate forms are there.
Okay.
When monohydrate forms are there, obviously it is in crystal structure forms or dihydrate forms. So we can't handle lithium hydroxide, sir. It is hygroscopic. So that's why.
That is a problem. Yeah, that's why
Yes,
I'm
sir.
asking.
Correct.
Yeah.
So molecule itself I have taken as lithium hydroxide monohydrate forms.
Okay.
So which is in crystal form.
But in the procedure, you have written only lithium hydroxide. That's why I'm asking.
No, sir, it's there. Yeah, sir.
Yeah, lithium hydroxide written. That's why I'm asking.
Yes. Actually, in scheme it's there, sir. Lithium
Yeah.
hydroxide.
Okay. Another one.
Sir.
Another one. How to calculate the ulcer score?
Ulcer score based upon ulcer mucosa layer streakings, like we'll give markings, sir. 1, 2, 3, like that. Markings will be given to it. So
okay.
any sort of streaks are there, we are getting 3 points like that. Based upon streaks format, we will select, sir.
What type of software you have to use it for drug likeness properties? Estimation of drug likeness properties.
Drug likeness properties, two softwares I have used here. One is Osiris Property Explorer, like that.
Okay.
So we can identify, this is online free software. Whatever the molecules that we are, smiles we are introducing, we are getting a data. So all the drugs, whatever I have selected, these are all obeying the Lipinski rule of 5. So it is showing good negative toxicity risks.
Lipinski means 0 to 5, I think, sir.
Yes, sir. Like molecular weight should be below 500. Bond donor.
Okay. Arif, anyway. Thank you.
Thank you, sir.
Thank you for nice presentation.
Okay, what is
Dr.
the
TG.
recommended dose of diclofenac sodium?
Sir, 100 mg per kg.
For human being.
Sir?
For human being, what is the normally used dose of diclofenac sodium?
Yeah, 100 and 200
Because in the
forms of.
market, diclofenac sodium is available in the form of tablets as well as injection.
Yes, sir.
So what is the normal strength of diclofenac sodium? Recommended dose?
Recommended dose is around maybe 200. I think so. Actually,
Sorry.
reinvented
The
that.
recommended dose is 50 mg in case of IR and 100 mg in case of sustained release tablets.
Yes, sir.
And assuming the adult body weight is 70 kg, then
Yes.
what is the rationale to select a dose of diclofenac 50 mg per kg, 100 mg per kg in your studies? The normally recommended dose is 50 mg per 70 kg body weight.
Yes, sir.
But in our studies, you used 50 mg per kg and 100 mg per kg.
Yeah.
So how you justify this? Because we have to use the dose which is normally recommended, normally used, but in our studies it is not so. What is the rationale behind it? Okay.
I can
Next,
get to that. Okay.
you used omeprazole, which is a proton pump inhibitor.
Yes.
And instead of omeprazole, why you can't try with NSAIDs? Because NSAIDs are having anti-inflammatory as well as ulcerogenic activity. So if you challenge with same NSAID, which is anti-inflammatory as well as ulcerogenic, like indomethacin,
Yes, sir.
that may be better instead of omeprazole, because omeprazole mechanism of action is different.
Yes, sir.
So can you justify this?
No, actually, I didn't went for indomethacin, sir. Actually.
Because indomethacin is having anti-inflammatory as well as ulcerogenic activity.
Yes, sir. Yes, actually. I would have
Then
went
how
that.
you determine the total polar surface area? Because in one of your slide you specified TPSA values.
Yes, sir. It based upon
Your
the
compound is having
polar,
good TPSA, more than 70.
based upon the electronegativity atoms, sir, like nitrogens, oxygens.
Means what are the functional groups present in your drug
NH,
contributing polar surface area,
NH,
and
NH
what are
groups.
the elements?
NH groups are there.
NH group.
NH groups are there.
Only NH group, because of only NH group, you got that 79.56. Because NH group
No, sir.
contribution is
Actually,
not much significant
NH
in our structure.
NH group is there along with P/1. Methoxy groups are there. OCH3 groups are there. When compared to P/5, no methoxy groups are there. So that's why it is somewhat less.
And what about onset of action, duration of action? Because in your compound, how much time is required to elicit the pharmacological activity and how much time that activity is observed? Similarly, whether you compared this onset and duration of action of your compound with either omeprazole or diclofenac sodium, because you observed statistical treatment. So statistical treatment of the data pertaining to either onset of action or duration of action, so that any significant differences between these pharmacodynamic parameters are existing or not.
I didn't check in that manner.
Because at different time intervals, definitely there will be differences
Differences,
that will
yes.
be statistically significant. So if you treat the data pertaining to onset of action or duration of action, that may be better.
Yes, sir.
So that is my one of suggestion.
Yes, sir. Sure.
Okay, anyway, thank you
Thank you, sir.
for your presentation.
Thank you.
Thank you, sir. Thank you, Arif Pasha.
Thank you.
Very good presentation. Next, go for next presentation. The name of presenter is Surajit Maiti. He was assistant professor, Haida University of Pharmacy, West Bengal. Surajit Maiti. Are you hearing me, Surajit? Surajit. So I think he was absent. Okay, next participant. T. Hemant Kumar, Associate Professor, Sri Kupa Institute of Pharmaceutical Sciences, Siddipet, Telangana. Hemant Kumar. Hemant. T. Hemant Kumar.
I think he does not join in this.
No, Subhay.
Sir, he is also absent.
Okay, we will close. Okay.
Maybe he is also absent.
Hemant.
No, sir, he is not in the participants list.
Hello, Hemant.
Hello, sir.
Okay, Hemant.
Sir, myself, Hemant.
Hemant.
Hemant P.
Hemant P. I think you are not.
I'm in Hemant, sir.
No. Hemant T.
No, sir, I'm in Hemant from Alareddy.
Okay.
Call for the presentation. Sir, we are completed the all presentation for this session. Thanks for evaluation, Dr. T.G.K. Murthy sir, Bapatla College, Dr. Rajendra Prasad sir, Viper College, R&D Director, Dr. M. Srinivas Reddy sir, Principal, Vageshwari College Pharmacy. Thank you, sir, for spending your valuable time for this session, and thanks for evaluation of all the presentations. Thank you, sir. And all the participants are requested. We will give the results after the discuss with the committee of the evaluation. We will give the best posters and announce at the time of validatory function of the second day of this conference. Thank you all.
Thank you.
Thank you very much.
Sir. Hello.
We can finish the e-poster presentation. Thank you all the participants and those who are presented e-posters. And I would like to thank you, T.G. Murthy sir, Principal of Bapatla College of Pharmacy, and Dr. Rajendra Prasad sir, Director of R&D Department, Viper, NASA Board, and Dr. M. Srinivas Reddy sir, Principal of Vageshwari College of Pharmacy, Vijayanagar. I would like to thank you, Dr. D. Kumar sir and Dr. Prasenjit Bendel Kumar sir for your valuable time. Thank you, everyone. And thank you one and all for your patience. And here I want to announce you all the participants.
The session will be closed. The one day, today's seminar session will be closed.
Best poster presentation will be announced in the validatory section tomorrow. Thank you all. Stay safe, stay healthy. Thank you. Namaskar.